In this presentation, we'll be discussing the gut microbiome and its integration with metabolomics. So the outline is as follows. We're first going to go over some very broad strokes in understanding the gut microbiome. Then we'll look at its deep connection to the host metabolome and assess strategies for basically profiling the gut microbiome, what technologies are currently available out on the market. We'll also go over strategies for improving gastrointestinal health, and then look towards personalized gut microbiome treatment. So first, before we really get deep into the gut microbiome, we need to understand its impact on health and disease states. Harbored inside the gastrointestinal tract at increasing concentrations descending towards the colon resides a collection of microbes known as the gut microbiome. In recent years, the gut microbiome has really emerged as an important, though highly complex, black box factor in relation to us, the host. The gut microbiome is compelling for its deep connection to human health and its tremendous taxonomic complexity and functional capability. Over the last 2 decades, as shown here in this Nature Reviews microbiology paper, we see its vast and encompassing connections with us, the host, whether it be from the gut-brain interactions with the gut-brain axis, the gut-liver axis, interactions with adipose tissue, interactions with the immune system, and also being extremely important in us developing as humans. Now, kind of looking over the history, it's important to first understand that even microbes themselves weren't really known to exist until 1677, and not really until the early 2000s, based on the advances in sequencing technology and computational technology, we weren't really able to do deep profiling of the gut microbiome. Of course, now with these advanced technologies, this field has really exploded, and we're uncovering vast microenvironments within the human, whether we're looking at bacteria, viruses, protists, archaea, and other microorganisms that are really fundamental to our health and our lifespan. Now, looking at the gut microbiome in adulthood, where most people are getting gut microbiome profiles done and looking to intervene with various different treatment modalities and dietary interventions, we actually see that the adult microbiome is actually fairly stable, fairly resilient. Now, as a broad proxy for the gut microbiome, researchers and clinicians will often look to different diversity metrics. Generally, it's assumed that the more diverse, the more rich your microbiome environment, the healthier the individual is going to be. And this ties into resilience, as if we kind of take these pictures here demonstrating the rainforest to your right, an increased level of diversity, ecological diversity, can basically handle a lot of different perturbations or insults to the environment being able to rebound, versus an environment that is very sparse in terms of different microorganisms, really not being able to handle those assaults, essentially being a more fragile environment. However, we do understand now that diversity by itself is not the best proxy for health, as we can have individuals that have a very diverse gut microbiome, but can be in a disease state or have different symptomology. A classic example would be constipation. Oftentimes we'll profile individuals that have constipation, and they'll also have high diversity. So really, this is one component that we want to assess when we're looking at the gut microbiome. We want to look at things, obviously, like the taxonomy, what type of microbes are there, as well as what kind of functions they're serving within the gut. Now, looking towards the blood metabolome, this is very important because obviously we're getting an assessment of what's going on with the host, particularly with what's going on in circulation. We know that the gut microbiome, while housed in the gastrointestinal tract, is not independent from the body. It's producing a lot of different metabolites that are actually coming into circulation and are interacting with us, the host. Now, looking at its overall biomass, it constitutes approximately 200 grams. This actually can fit in the cup of your hands. It's the reason I have that image there. But it's estimated that approximately half of all the metabolites in our body have been synthesized or modified in some way by the gastrointestinal microbes. Therefore, the assembly of microbes in our gut are really tightly intertwined with our health and, importantly, represent an important therapeutic target. The microbiome can really serve as a critical connector and communicator, interacting with various omic layers, even like the genome, the proteome, looking at proteins, transcriptome, and of course the metabolome, to really offer a comprehensive view of human health and disease. And just a few key functionalities of the gut microbiome, obviously based on its location in the gastrointestinal tract. It's helping harvest food energy, can improve gut function, can reinforce gut barrier function. It's very important when we talk about interactions with the immune system. In a similar fashion, it can help protect against pathogens. It can also produce very important metabolites for our health, like short-chain fatty acids and modification of bile acids that we'll discuss. It can also synthesize vitamins and hormones, boost the immune system, influence organ systems, and even affect our metabolism of lipids and glucose. So directing our focus to short-chain fatty acids, these have long been appreciated as the primary vehicles for energy salvage. Now, recent research has actually shown that short-chain fatty acids possess potent signaling functions that modulate energy intake, energy utilization, and inflammation. High-fat diets with or without short-chain fatty acid supplementation have been reported to have inhibitory effects on weight gain. So actually, we see some anti-obesity application. Host metabolites, including bile acids and immune factors, also interact bidirectionally with the gut microbiome and also influence its contribution to energy balance. Butyrate, a short-chain fatty acid, actually enhances thermogenesis and energy consuming brown adipose tissue, whereas other short-chain fatty acids like acetate and acetate-rich short-chain fatty acid mixtures actually increase white adipose browning. So here we can actually see some phenotypic changing that is going to translate in somebody having a more lean-associated phenotype. Propionate can promote gluconeogenesis in the liver and the intestine, basically the formation of glucose, whereas hepatic acetate has been reported to reduce lipogenesis, limit fat accumulation, increase the expression of thermogenesis and fatty acid oxidation genes, and reduce inflammation. Moreover, a given short-chain fatty acid compound can influence physiology in opposing ways in different tissues, as exemplified by acetate or butyrate supplementation, increasing different receptors in adipose tissue while reducing it in the colon. So such differential effects raise the possibility that metabolic states could be manipulated via specific short-chain fatty acids. Now, when we feed these microbes the proper substrate, they can actually start producing a lot of these short-chain fatty acids, which can interact with different tissues in our body, like adipose tissue, to actually shift our health state. Another essential signaling pathway to consider is the metabolism of bile acids. Primary bile acids are exported into the bile, where glycine and taurine are deconjugated by the microbiota that express bile salt hydrogenases. Now, accumulating evidence suggests that bile acids can regulate muscle metabolism by regulating the activation of bile acid-specific receptors like farnesenoid X receptor and transmembrane G protein coupled receptor, TGR5. These processes are fundamental for enhancing lipid and glucose metabolism. Furthermore, increasing the population of microbes that express high levels of bile salt hydrogenase, such as common examples like Bifidobacterium, Lactobacillus, can alter bile acid synthesis in the intestine. This alteration holds tremendous potential for the strategic use of diet and fermented foods. Looking at other interactions beyond the liver and adipose tissue, we can also look at central nervous tissue. Microbial metabolites can alter feeding behavior, as exemplified by the exogenous delivery of short-chain fatty acids, reducing appetite. These effects are thought to be mediated through the gut-brain axis, a signaling network linking the central and enteric nervous systems. Short-chain fatty acids affect energy intake by signaling through the vagus nerve, or in the case of acetate, by actually crossing the blood-brain barrier and inducing the expression of anorexia-promoting genes. So here we see direct effects from microbial products on brain tissue. Many gut bacteria synthesize peptide mimetics of hormones regulating satiety and hunger, such as leptin and insulin. Gut microorganisms can also synthesize neurotransmitters, such as GABA, dopamine, acetylcholine, and adrenaline, as well as affect endogenous levels of serotonin in the intestine and dopamine, noradrenaline, and serotonin in the brain. These neurotransmitters can then affect energy intake through their roles in gut motility, satiation, and food reward. So clearly there's tremendous application here with assessing the gut microbiome and looking at how it's affecting host physiology. And as I alluded to earlier in the presentation, it really hasn't been until more recently, in the last few decades, that we really had the technology available for assessing the gut microbiome at a very deep level. And there's different tiers of assessing the gut microbiome when we look at, say, sequencing strategies. So looking at some more basic stool tests, we know, of course, there's tremendous clinical use for fecal occult blood tests. We might do immunochemical tests, stool cultures. But really, I think the future lies in microbiomics or sequencing strategies. We can take some other broad proxies, of course, breath tests, blood tests, even imaging studies, as well as basic clinical symptomology scales, which we actually do use, the GSRS, the Gastrointestinal Symptomology Rating Scale, which are valid, but really they need to be integrated with these more complex and advanced technologies. So really, I would say the workhorse of microbiomics for a long time has been looking at ribosomal RNA, which we term 16S because it's targeting the 16S region of ribosomal RNA. When we look at the structure, we see that there are what we term hypervariable regions. There are 9 that we can profile. Basically, these are, when we say hypervariable, they're different across different taxa or different microbial features, and that can help us annotate which microbes they are when we base it against a library based on the RNA. And this actually helps us decode all of the material that we're sequencing. Generally, when we're doing 16S for humans, and actually it can expand that to mammals, we're generally looking at the V4 region. This, you can usually get resolution down to the genus level. So if we're looking at the tree of life, we know, of course, we kind of come down from a kingdom level. We might go down to class, family, genus, species, and then strain. So there is a limitation with 16S sequencing in that we're kind of looking at a higher level at the genus classification. There are some newer technologies that do what's called a long read, looking at all 9 hypervariable regions. and their claim is that they can get down to the species level. However, they're not going to compare with deep sequencing, which we'll get into in a second. So this is just showing you the hypervariable regions that you can see there in the top panel to your right, looking at all 9 of the hypervariable regions. And again, I'm just reinforcing that you're just getting down to the genus level in terms of resolution. There are different methods that you can use. You can tag different ribosomal RNA. Generally, when you're doing 16S amplicon sequencing, you're looking at bacteria. However, you can look at prokaryotes. You can also, with a different tag, you can look at eukaryotes with 18S ribosomal RNA. And for fungi, you can also do a targeted internal transcribed spacer. However, when you're doing these analyses, these are additions to the 16S sequencing and are generally not included when you're using this technology. These are really kind of add-ons. So the future really lies in what we call shotgun sequencing. So this is where we're targeting the whole genome. We term it metagenome. Okay, we're not just looking at one gene because, as we know with the microbiome, there's thousands of species that we can profile. So what we do is we get a fecal sample in the case of the gut microbiome. We extract the microorganisms and we break down the cell wall and we pull out the DNA, not the RNA. So this is an important distinction. We then break down the whole genome, all the DNA that we collect, into these short random fragments, and then we sequence those short random fragments. The really intensive process here bioinformatically is then assembling all these short little reads into long contigs. You can almost kind of think of this as putting together a puzzle. You have all these little puzzle pieces that represent those short little fragments, and then you're building out all of these long contigs, which we can then assemble and rebuild as a whole genome, and then annotate not only what they are taxonomically down to the strain level, but also what functions they're performing, which is a very important distinction in comparison to 16S amplicon sequencing. So just a quick side-by-side, generally 16S sequencing is going to be in it more less expensive in comparison to shotgun metagenomics. However, you are going to be generally limited to the genus level. In comparison, shotgun metagenomics, you're getting down at the species and the strain level, and you're also annotating functions. You can do some inference with 16S amplicon sequencing, but there is an error rate associated with that, and generally it's assumed that you're going to have about 80% accuracy here, and you're going to be missing a lot, right? Because we're not getting down to the deep resolution as we would with shotgun sequencing. In addition, you're missing out on a very important kingdoms that we know are in the gut microbiome, such as plasmids, phages, and viruses. Now, the cost here is not only due to the type of sequencing that we're using with shotgun metagenomics, but also downstream, the computation that's involved really profiling this can be time intensive and obviously eat up a lot of computational resource because of the mass amount of data that we're getting from these sequencing reads. So there are things beyond when we're assessing the microbiome, just the metagenomics. We can also profile metatranscriptomics. We can look at metaproteomics. We can also look at metabolomics in the gut microbiome, because just like the host, we know that, if we take a systems biology paradigm, in our own bodies, we have our genome, we have our transcriptome, which is going out from the nucleus of the cell, interacting with different organelles. And then building proteins, right? So we have the proteome, and then all the functionalities of the proteome can translate into metabolites. So the same thing is occurring in the microbiome, and you can assume a full systems biology paradigm in that environment as well. So strategies for improving gastrointestinal health. I think because of the black box nature of the gut microbiome, a lot of people, they first kind of think about probiotics or prebiotics when they look at um, addressing gastrointestinal health. But there's some important caveats that we need to understand when we're giving any type of intervention directly targeted at the microbiome. For instance, the gut microbiome is colonization limited, meaning that we can't just take a bunch of probiotics and assume that they are going to take up residence and start proliferating. You have a pretty stable gut microbiome configuration that in many cases is going to be very resistant to exogenous microbes. It's also important to understand that constitutional bacteria are not present in all individuals, just waiting for the right environment to grow. Our microbiota is really constrained by the type of bacterial colonization that we can effectively have throughout our life. Now, there are opportunities to essentially augment adaptation with targeted dietary and supplemental therapies like the probiotics, the prebiotics, and even the postbiotics. But we have to understand that it's very unlikely that we're going to completely change all the taxa in our gut microbiota. In fact, when we look at how unique microbiotas are across individuals, it's essentially as unique as your fingerprint. Everyone is going to have a very different microbiota. That's not to say that we shouldn't intervene if someone has a configuration that is associated with disease. For instance, in this study conducted at Arizona State University, looking at college students, basically freshmen first coming into college in dorms, we see that certain profiles may indicate metabolic imbalances associated with things like prediabetes, cardiovascular disease, and other chronic conditions. So if we can detect a disease-associated profile, this kind of gives us an early warning sign and allows time to intervene, whether we're doing lifestyle modifications, dietary changes, or personalized treatments to prevent or mitigate the progression of diseases before they become clinically apparent. So for example, once in adulthood, the main moderator of the relationship between us, the host, and the gut microbiota is diet. Metaphorically, the gut microbiota often assumes kind of a co-pilot role within the host. Indeed, a perturbated or disturbed gut microbiota could be engaging in compensation type actions aimed at conferring host health benefits. Therefore, microbiota data needs to be placed within the context of the host and should be complemented with other host-centric omic layers, such as transcriptomics, and really for us, blood metabolomics. So with this particular data set, we looked at the microbiome and we also looked at the metabolome. And what we found was higher ratios of a genus of bacteria called Prevotella and another called Bacteroides correlated negatively with P-cressyl sulfate and cholesterol sulfate, implying potential health implications while positively correlating with a neurotransmitter. Now, these are important because P-cressyl sulfate, a microbially generated protein-bound uremic toxin, is associated with kidney disease progression. So here we're already seeing in these essentially late-stage adolescents production of detrimental metabolites. So what we're wanting to do is actually shift these ratios of bacteria that we were assessing Interestingly, Prevotella, low levels of Prevotella, which was contributing to this increase in p-cressyl sulfate, is associated with higher fiber diets. And when we looked at the diets of these individuals, they were not consuming adequate intakes. So a very simple solution over time would obviously be to have these individuals consume more fiber to correct these metabolic profiles. Now, diet, of course, is a pretty contentious topic. In our modern food systems, we know that there are some things that are kind of omnipresent. I mean, a good example that I'm showing here in the top panel is emulsifiers and non-caloric artificial sweeteners. And these have really become ubiquitous in industrialized diets. I mean, you can do your due diligence and avoid them, but it's very easy to consume a lot of them if you aren't really paying attention. Now, these are generally, according to the FDA, they have GRAS status, meaning generally recognized as safe. However, as I'm showing here, recent studies have demonstrated that these compounds impact the gut microbiome and its contributions to energy metabolism. Emulsifiers, common ones like carboxy, methylcellulose, and polysorbate 80 have been observed to disrupt the intestinal mucosa, essentially the epithelial cells that are lining and protecting our gut barrier, leading to microbiota encroachment on this tissue, causing low-grade inflammation, adiposity, and high blood glucose levels that that were transmissible in a preclinical mouse model via microbiota transplantation. So, while recognized as safe, clearly that's not the case when we're talking about the gut microbiota and spurring low-grade inflammation and metabolic derangement. Now, alternatively, the consumption of fermented foods has been shown to increase microbiota diversity and decrease inflammatory markers. So someone coming off these types of diets, with high consumption of emulsifiers and various dyes, there are dietary solutions, while perhaps not as, I suppose, easy as taking a dietary supplement. They are very potent in reducing inflammation markers and reconfiguring the microbiota to something more health associated. Another major issue, of course, that I want to bring up that we capture when we assess the microbiota is SIBO, so small intestinal bacterial overgrowth. The prevalence of SIBO, both in patients with irritable bowel syndrome, IBS, and controls, was found recently to be very, very high in populations in the West. For instance, the United States, we're looking about 55% of individuals with IBS having SIBO, and in the controls, 37.2%. So very high prevalence. And a lot of people may have this and not even know it. This is a condition characterized by an abnormal increase in the bacterial population in the small intestine. Basically, certain bacteria that are encroaching on a segment of the intestine that it shouldn't really be there. Normally, the small intestine contains relatively few bacteria compared to the large intestine. In SIBO, the bacteria are typically found in the large intestine, begin proliferating and growing in these regions, leading to various digestive symptoms and potential complications. Now, there are many treatments available. A lot of physicians will first turn towards antibiotics. There are dietary modifications that are pretty powerful in mitigating this issue, particularly assuming a low FODMAP diet, or perhaps an elemental diet, which really needs to be done under medical supervision, and also the elimination of refined sugars and processed foods. There are also other strategies, again, probiotics, prebiotics, digestive enzyme support. However, a very interesting area that we have done research in is using intermittent fasting. So for this type of strategy can actually vary the amount of time that you're essentially not consuming any calories, whether it be liquid calories or foodstuffs. Now, the interesting thing about fasting is it promotes what we call the migrating motor complex, which is crucial for sweeping bacteria and undigested food from the small intestine to the large intestine. This process occurs about every 90 to 100 minutes. But when you're fasting, this is only occurring when you're fasting. So if you're eating a normal breakfast, lunch, and dinner, you're in a postprandial state a lot of the time, right? You're processing food, and you're not experiencing the migrating motor complex very often. So when you're fasting, you're actually allowing this process to occur and kind of reboot the system, if you will. Additionally, when you're going for various fasting lengths, when someone's assuming a regimen, meaning that they're constantly doing this for weeks or months on end, you can have the increase in anti-inflammatory bacteria like the lactobacillus and bifidobacterium that we commonly see in probiotics. So there's actually a natural way to do this without ingesting those microbiota. We also see a reduction in different pathogenic bacteria. And I will call attention to a very interesting microbe that generally proliferates during this period. It's called Akkermansia muciniphila. This is also now has been commercialized and as a probiotic. Essentially, this is considered a steward of the mucus layer in our gut. This promotes the increase in healthy levels of mucus, and this offers a buffer against the luminal contents of the microbiota interacting with the brush border. So with this increase in this microorganism, we can actually see decreases in inflammation in circulation. So probiotics, of course, are very, very, very popular. Data from the U.S. National Health interview survey show that probiotics are actually the third most commonly used dietary supplement amongst adults. They're widely available. You can find them in capsules. You can find them in gummies. You can find them in yogurts, fermented foods, of course, kefir, kombucha, various elixirs, and even food products like sourdough bread. The interesting thing about probiotics, however, is when you think about the actual quantity you're taking, you can look on the back of your product and see that, a good probiotic, a multi-species probiotic is going to have, say, anywhere from maybe 10 to 50 billion colony-forming units or CFUs. That's really a drop in the bucket when we look at the overall biomass in the gut. And one of the main mechanisms of action of probiotics is actually through interacting with the immune system. So to put that into perspective, about 70% of our immune system resides in and around the gastrointestinal tract, constituting what we call the GALT, which is gastrointestinal associated lymphatic tissue. If we look at the overall surface area of the gastrointestinal tract, we're able to map it out across the floor, including all the villi and the crypts and all the little spaces. We're looking at 200 square meters, which translates in feet to over 2,000. So this is really on par with a single family home. So it's tremendously complex, massive, and has immune tissue everywhere. It's fairly safe to assume that unless probiotics are targeted, and we know that you're going to have a microbiota that's conducive to them actually grafting and interacting with the microbiome in that tissue, that they're going to do something, right? So if we're taking things indiscriminately, the likelihood of them actually doing something is fairly low. They really need to be targeted. And with the microbiome in general, if we're trying to target shifting the configuration of the microbiome, all treatments, I'm of the opinion that all treatments really need to be targeted. Otherwise, we're just kind of wasting their intervention. Now, in terms of interventions and really what's going to move the needle, as I said earlier, the gut microbiome is very resilient once you come into adulthood. And there are very few things that can really shift it. We know that antibiotics, of course, can do that. I would advise against indiscriminate use of antibiotics. I know there's been some talk recently about using antibiotics in a targeted fashion to decrease pathogenic microorganisms. But really, we need to be very careful when we utilize those because in many cases, they can have collateral damage in the microbiome. I think in terms of shifting the needle, something that is going to be more long-term and actually over time is going to have a greater effect is really going to be a specific diet. Other things that may not be as sexy, of course, are going to be exercise, all the normal things that we advise, people to live a healthy lifestyle, sleep, staying hydrated, moderating medication use that can affect the microbiome, limiting processed foods and sugars, which is actually huge and obviously ties into diet, incorporating microbial accessible nutrients, or MAX is commonly called for fermented foods like kombucha, obviously sauerkraut. You can do different types of fermented milks, things like that. And really probiotics and targeted use of probiotics and prebiotics in their current preparations are pretty low on the list. They can be used in a targeted fashion, as I mentioned earlier, but the main levers for action are going to be the things that aren't as sexy. Now we'll focus on looking towards personalizing gut microbiome treatment. So looking at the systems biology paradigm, of course, with the host, we look at the blood metabolome, mainly because it is a great barometer for overall metabolism, and this is something that can change pretty rapidly. So if we have an intervention, we can start to see shifts in that person's metabolism and adjust from there. Other omic layers are not going to have that type of longitudinal expression, particularly the genome, which really should just be done one time if you're doing it with accurate and good depth. Transcriptome, that is done at a lesser cadence than the metabolome, as well as the proteome, which is going to be a bit more stable. The microbiome is similar to the metabolome as it can shift quite rapidly. So again, this is a very attractive target when we're assessing health. And if we're targeting the microbiome, this is something that we can do quite frequently, and then we can form health trajectories for that individual. So this was a study that we published in Nature Communications in 2024, where we looked at the effects of two different types of calorie restriction. One was a normal calorie restriction model that was based off of USDA dietary guidelines, just using a continuous approach where calorie was restricted to 25%, where the individual consumed breakfast, lunch, and dinner 7 days a week. The other dietary arm was looking at intermittent fasting. So here, individuals were fasting 1 or 2 days per week over the duration of the study. This was a shorter term study because compliance is actually fairly difficult when you're doing these types of studies, not only due to the caloric restriction or the fasting that you're imposing, but all of the sample collections that we're imposing on these individuals. However, in just that short amount of time, we were able to detect very interesting interactions, changes in the microbiome, and then changes that reflected in the blood metabolome. Looking at these co-occurrent features, we found that in the fasting group, there was metabolites associated with protein degradation and really robust mobilization and oxidation of fatty acids. And this was reflected in just basic chemical assays, and also looking at DEXA scans, where the individuals in the fasting group lost significantly more overall adiposity, with large amounts actually coming from the visceral the visceral adipose tissue. Interestingly, we know that every microbiome is different. And when we look at the response to these different types of interventions, in the case of this study, we saw a pretty wide variation in weight reduction response, as you can see here to the left. I mean, generally we assume that a clinically significant weight loss is about 5% of total body weight. While many individuals did achieve that, they paled in comparison for several individuals that lost, in some cases, double or triple that. And this is, of course, looking at relative weight loss. So, regardless of the person's initial weight, some individuals just responded very, very well to the fasting regimen. And we looked at the response in the microbiome and the metabolic profiles. We saw some pretty stark differences. Notably, when we looked at the fecal metabolome, those that really responded well in the high weight loss group had an enrichment of fecal metabolites involved in lipid metabolism, whereas those in the low response group had increases in pathways related to amino acids and peptides. So in the high weight loss group, their microbiome configuration was really supercharged for handling and disposing of lipids. So again, it's very important to understand someone's initial composition coming into an intervention. That way that we're not wasting time and we're going to find the most effective strategy. And we looked at one individual that responded very, very well, in fact, the best to the intermittent fasting diet. And we continued this study. We had an agreement with our IRB, Institutional Review Board, to continue this study for a year follow-up. And over that year, this individual lost 100 pounds and totally reconfigured their microbiome and kept the weight off. So with the personalized recommendations that we provide this individual for the year that we tracked them, we actually saw that, again, one of the major issues with high responders and weight loss is they might initially lose a lot of weight, but then they'll have recidivism and rebound. We get this yo-yo effect. With highly specific interventions, tailoring not only the diet to configure the microbiome, but also work with the person in terms of different behavioral aspects, we can have very, very successful outcomes, such as in the case of this individual, and then reconfiguring their microbiome, which translated into metabolism that was associated with a leaner phenotype and reductions in appetite via different hormones that we profiled. So, in summary, the gut microbiome is essential for health. It's considered a black box factor, and in many cases, it's neglected, or when it's assessed, only a few features are looked at, which is really doing a disservice to all the data. While it's generally small in terms of biomass, it has tremendous interactions with us, the host, whether we're, in the previous case that I was showing, causing changes in our metabolism, if we're going to better mobilize fat from our tissues, synthesizing vital vitamins such as B vitamins, vitamin K, as well as hormones, particularly appetite-associated hormones. And then, of course, boosting our immune system, which translates into quelling inflammation in our body, which can affect every organ. There are different analyses available. I would recommend, if you are going to profile the microbiome, looking into deeper sequencing. 16S, while it's inexpensive and cheap, this is not something that I would really hang my hat on if you're really interested in getting a full picture of the microbiome. And in terms of interventions, the microbiome is very responsive to changes. We can cause changes here if the changes, if the interventions that we are providing someone in line with their overall composition. And a lot of these are going to be dietary. So I advocate for the profiling prior to intervention and then the follow-up over time to really assess that person's microbiome, other health states, and see if they are receptive, the microbiome is receptive to that therapy. We also recommend the integration with other features, whether we're looking at clinical blood work or which we feel is going to be revolutionary in this space, the metabolome of the host. So with that, I will end.